Okay, good morning, everybody. Can you hear me? Fine? Yes, fantastic. So, thank you, Stephen, for giving me this great opportunity. And I'm really excited and enthused because more than anything else, I consider myself a science storyteller. In fact, I used to write a science column for my local newspaper. I used to call it Science in 10 Minutes. We will talk more about that later at an appropriate time. But today I'm here to not give a technical talk, but rather do what is called an episodic history and tell you the story of an organism. So, as the title says, Agrobacterium, the biography of a natural genetic engineer. And you will see how it is going to really give a new kind of perspective as to what we do with research and how we keep ourselves excited enough to do great things in science. First off, uh, I want to dedicate this talk to a great legend of a personal uh, hero of mine. Her name is Mary Del Chilton, and as you can see, from an unassuming little girl, she grew up, evolved herself to build her own telescope. And when she goes out into the field, that's she with the whole family, the husband and the little kid looking ahead, an eager young mind, while she is scouting everything in nature. She went on to win the World Food Prize and the Presidential Medal for Science, if I'm not mistaken. So, a very great personality. And pretty much this talk today is because she wrote she wrote a wonderful article, which I'll come to at the end of the talk. I'll give you that reference as well. But this is what enthused me into giving this talk as well. So, what I do for a living, for the past 3 decades, started off way back in 1991. I started my life as a plant scientist. I did tissue culture in the beginning, and then I evolved myself into a transgenesis of crops. So I'm a certified plant genetic engineer. And along the way, I treated my research as not just going after the products of plant genetic engineering, but what I realized is we need model systems to engineer and understand our environment. That's what my premise is always. We learn from nature. And when we do that, I have been doing plant genetic engineering for the past 3 decades, as I told you, 25 years of it in academic settings. And just last year, I made a move to the corporate industry. My friends keep calling that a very gutsy move, leaving tenure. But I left it because I was losing excitement in my scientific life. And so I made the move. So I started my life in United States at Texas A&M University at the Norman Borlaug Center for Southern Crop Improvement. And then I became a assistant professor in 2010 at Western Kentucky University, and I got my tenure, stayed there till 2023. Now I am with a startup in Sacramento, California. It's called MyFlora DNA, where I'm working with CRISPR editing of crops. So more about that later before I come to what I would like to say I took for granted, right? All along, I have been worried about reshaping plants and getting better plant products or talking about plant traits, but I never paid much attention. I did, but not to the extent I would have liked. Is this little organism called Agrobacterium. I took it so much for granted. Well, quietly in the nature, it was actually doing its job. Even before scientists went after it and reprogrammed it to do what they like, it was a natural genetic engineer. And when you really look at it, a natural genetic engineer, what did it do? It wanted to make something for its own benefit. Okay, and we'll come to that in a little bit as to what it wants from the nature, right? But it went to a plant, it occupied a plant at the cellular level. So it went to a plant cell, it cut a part of its soul, right? That's what I call the DNA. DNA is the best definition as far as soul is concerned, right? Whenever I do a course of non-majors in my one of my favorite courses to teach is biology for non-majors. When I have a room full like 300 students of everybody not doing biology, they are from music, philosophy, physics, chemistry, what have you. I tell them that before you die, you've already taken half your DNA into your kids, right? So that part of your DNA has been leaving you, your parents, your great grandparents, whatnot. So half of you is already out. So what is it that passes on to the next generation is your DNA. So when we look at all our scriptural texts, they say you come and go, but your soul keeps moving on. What's the best scientific evidence for that? It's a soul. That's the DNA, right? So this agrobacterium is cutting part of its soul or the DNA, and it is injecting it into the plant cell, where it not only takes care to the channel where that DNA gets into the plant, it also protects itself from the plant nucleases, meaning it protects itself from getting degraded. It gets imported into the nucleus and it becomes part of the plant. And this has been going on in nature. And there were a group of scientists who actually discovered this and then harnessed it to be a tool that's brought about all the revolutions in plant genetic engineering. What about this natural genetic engineer, right? We need to look at its life story, where it started or where it was discovered, right? And how it laid the foundation of plant genetic engineering. So that's what I've been taking for granted as to where did it actually start? Who was the first person who actually got into the crux of this whole thing, right? So the story starts with, in all earnest as to where this thing was discovered first, it was in 1897. So that was because of a scientist by the name Fridiano Cavara, right? He observed these grapevine galls, abnormal tumorous structure on grapevines. They were also referred to by the pathologists as the wraths of grapes, right? So I get a kick in telling you that if there was a book called The Grapes of Wrath, which won the Nobel Prize, right, for John Steinbeck, we plant molecular biologists have won our own Nobel with the wraths of grapes. So that's a little bit of fun satire I would like to do. But Fridiano Cavara was the first one who observed these structures. Right? And then comes two great legends. One is Irwin Smith, who for the first time he published a paper where he compared these crown galls, as you call them, right? But crown galls, he compared them to the sarcoma in mammalian systems. And he wrote a paper, Crown Galls and Sarcoma, in the 1980s, I guess, 1907, 1917. And there was the discoverer of the plant hormone auxins. His name was F.W. Wendt. He said those tumor galls are actively dividing cells which are due to excess auxins. Where do auxins come from? The plant itself has a very judicious mixture of auxins which divide its cells and cytokinins which differentiate a plant into stem, leaves and roots, right? So a proper auxin will never give those tumorous growths on plants. So he said these are excess auxins which are an external source, right? And that's when we meet Armin Brown, 1926. He is referred to as the godfather of crown gall. So Armin Brown was the first person who actually removed those tumors or the crown galls from those grapevines, those abnormal structures, and he cultured them. And when he cultured them on petri dishes, he had like even without, there is a very popular auxin called 2,4-D, right? 2,4-D is also infamous in terms of being used as a bio control agent or a biopesticide sometimes, but that actually divides cells very actively, right? So 2,4-D, then there is the IAA, which is again another hormone of plant, and kinetin, which actually is a cytokinin which differentiates those same abnormal mass of cells into stems, leaves, and roots. So you can get to any part of the plant, take a part of the plant, put it on 2,4-D, first to get a actively dividing mass of cells called callus, and when you judicially mix it with cytokinins like kinetin, you can get back a whole plant. So the totipotency, as they call it, any part of the plant can become a whole plant. But what Armin Brown showed is these crown galls, without any addition of auxins, could grow on their own. So he said that that particular thing which is causing those abnormal growth of those crown galls on plants, he called it a tumor inducing principle which a bacterium is providing the plant. So basically that was a very, very revolutionary thought. So in fact, Armin Brown used to maintain these crown galls for time immemorial. He had those cultures in his lab. So that's why we call him the godfather of crown galls. When he actually removed a principle, and he showed it's an autonomous agent, not the plant doing it, but a plant in an endosymbiotic relation with the bacteria. Then comes Georges Morel in 1970. He actually said that the bacteria is getting into the plant, actually these amino acids called opines, and he listed out an octopine and nopalene. These are amino acids which are not found in nature. The bacteria is utilizing the plant's apparatus to make these opines. So this is the first instance, greatest eureka moment in plant biology, where you showed that a bacterial gene got into a plant cell and it functioned there. So it cannot make these opines on its own. So it gets into the plant and it hijacks the plant's machinery to make its own opines. So that was a amazing eureka moment for plant cell, which was discovered in 1970. Right. So now the job is to identify the bacterial DNA, because anything that happens in nature, there should be a piece of DNA which makes a RNA and which makes a protein, right? So that is what sometimes it is referred to as the central dogma of life. Anything that's happening in nature can be traced back to the DNA. So what is this bacterial DNA? What does it do? So we had three great scientists who spearheaded this revolution. One is Robert Schilperut, the other was Eugene Nestor, and then our own Queen Mary, as I would like to refer her to, Mary Del Chilton. So these three amongst themselves started identifying what this bacterial DNA is all about. And what they found, or at least there was another group which was spearheaded by some of the legendary scientists, Mark von Montagu and Jeff Shell in Kent, in the European Union, the Belgium, I guess. So in their lab, there was a student by the name Ivo Zenin. He discovered that this bacteria had an independent molecular unit, or what we call a plasmid. So every bacteria has its own DNA. Any bacteria that doesn't have any nucleus, so its DNA is floating around, we call it the chromosomal DNA. But apart from that, there are bits of circular DNA within the bacteria which are called plasmids. These plasmids can jump ship. They can go from one bacteria to the other. Sometimes they go from one organism to the other as well. But this particular plasmid, which is called the TI plasmid, TI stands for tumor inducing. It's inducing tumors in plants, right? So what Ivo Zenin, when he kind of deciphered its molecular structure, he could see that the TI plasmid, as you can see from the slide, has a particular small region called tDNA. tDNA, T stands for transfer. That bit of DNA is transferred into the plant. So that's what is called the tDNA. That actually contained a element, a gene, a piece of DNA which codes for auxins, which are these hormones which actually give those tumorous, abnormally dividing cells on the plants. And it also has, the plant already has auxins, and this transfer DNA is giving excess auxins. So that's a hallmark of why you see those crown galls. And then you have cytokinins also. There is a gene there. And then comes that last bit of piece, the opines. So the transfer DNA, when it is cut from precisely from this bacteria and enters the plant, it makes auxins, it makes cytokinins, and it makes opines. So what the scientists saw was, if this is in such a beautiful precision, if it can cut itself and get into the plant and make auxins, cytokinins, and opines, let's keep the bacteria happy. Let us not mess around with that opine. But since it doesn't require the auxin and cytokinin, why don't we cut those pieces out and put in whatever we like, right? So we can make that Agrobacterium happily reside in the plants, but we will also provide it with our own genes of interest so that it gets into the plant and introduces those genes into the plant, and we both are happy. So Ivo Zenon was the person who discovered the gigantic Ti plasmid, right? And then comes these two groups from across the Atlantic, one in Seattle, one in Ghent, who closed down on that tDNA. And as you can see on the left is this Seattle group, as we call it. So there we have Don Merlo, starting from the left, Martin Drummond, then you have Eugene Nestor, and then we have Daniela Shiaki, and then our own Mary Shelton, who played a very major role. And then we have Alice Montois and Mick Gordon. I don't know, in the audience, I might have some people who actually know these people. Mary Del Chilton herself was in Syngenta, and I have a kinship with Syngenta because as a grad student in India, I got a fellowship with Ciba-Geigy. Ciba-Geigy used to be the name of the company from Switzerland, which later became Syngenta and then Novartis and this. So Seattle group, right? So, and on the other side of the Atlantic, we have the group headed by Jeff Shell and Mark von Montagu, then Patricia Zambrisky and Ken Wang. These two groups in 1983, they deciphered the entire molecular elements within the Agrobacterium, and which I'm going to kind of detail a little bit to see how beautiful this thing happens in nature. How this precise transfer DNA is cut and it is sent to the plant and it gets integrated. So I would like to call 1983 the Agrobacterium annus mirabilis. Just like we had a patent clerk in Switzerland who sat in a patent office and wrote those 4 great papers of photoelectric effect and Brownian motion and the general and special theories of relativity, and we now can't take 5 steps in the whole world, right, without running into a discovery of that annus mirabilis set of papers. I'm talking about Albert Einstein. So we live in Albert's world. But just like that, now, as far as plant science is concerned, we cannot take 5 steps in any plant lab without running into a discovery that was made by the Seattle and Ghent groups. So that's why I call that plant genetic engineering 1983 as the Agrobacterium annus mirabilis. So, what is it about the Ti plasmid that you are looking at, right? Let's kind of zoom in on that Ti plasmid, right? What you see there is, you see those beautiful green triangles where you see what is called a left border and a right border repeat. That is precisely what is cut, okay, of the plasmid, and it enters the plant, right? And if you look at it, I will break down every element there. Right out of the right border, you have what is called the conjugal plasmid transfer. So the Tra gene, as the name says, Tra is transfer. So that codes for how that piece, once it is cut out between the left and right borders, it goes into the plant. The Tra gene is responsible for that. And a lot more, not just the Tra gene, but that is what you're looking at. Then you have the octopine catabolism gene. That is what the bacteria is aiming at. It gets into the plant to hijack the plant's machinery to make an opine, right? And then you have another opine gene, And then you have, like every plasmid DNA, there is an origin of replication, meaning this DNA should get into the plant and start making copies of itself, a perpetual photocopy machine kind of a thing. So that's the switch there. And after that is the business unit of the whole element. It's called the virulence region, right? It's called the vir region or the virulence region because these genes, the set of genes here, all the way from A, H, C, D, E, F, H, every bit of this virulence region is what does individual functions when the tDNA is cut and it is getting into the plant. And I'm going to list that. On the right hand side of the slide, you see that very paper from Jeff Shell's group, which was titled The Supercoiled Circular DNA in Crown Gals, which induces the Agrobacterium into doing what it does. So this is the beautiful molecular machinery which I'm going to list out now, right? So on the slide you see the plant cell, right? That hexagonal plant cell, and then you have the circular bacterial cell. What happens, right, when the Agrobacterium goes and bites into the plant, right? I mean, I'm using very colloquial terms here, but yeah, that is just to make it very, very explanatory. So the bacterium goes and bites. As soon as it bites into the plant gives out these compounds called phenolics, right? The phenolics are the ones which are given out of the plant, and what it does is when the phenolics come out, within the bacterium there is this gene called CHVE. Right on top you see that where it says sugars. That CHVE, CH stands for chromosome, V stands for virulence. So it is actually encompassing a virulence, meaning it is each one is trying to defend its own cell, right? But what happens is as soon as the CHVE piece of DNA gets activated, there is a gene called Vir-A. So in that plasmid where I showed all the Vir genes with all the alphabets, the Vir-A is what gets first activated. That Vir-A gets activated because of the phenols that are coming from the plant, right? So what does it do? If you take the life's processes, the most important process is always a phosphorylation, addition of a phosphate element, right? So that's why we have these enzymes called kinases. If anything has to happen in our body or a plant or a bacteria, the first element that gets activated is the phosphate. So over here, a phosphate group, the Vir-A, causes a phosphate group to go and phosphorylize the Vir-G. So see the sequence now. Phenolics activate the sugars. They switch on Vir-A. Vir-A adds a phosphate group to Vir-G. As soon as Vir-A adds a phosphate group to Vir-G, that sets an avalanche of reactions. This Vir-G first activates the Vir-D gene. So Vir-A to Vir-G to Vir-D. What does this Vir-D do? It goes and cuts the transfer DNA. So from the TI plasmid, on the left and right borders, you have the Vir-D splice out that tDNA or the transfer DNA. As soon as that tDNA is cut, the Vir-D1 is what is called as an isomerase. What it does is it cuts the right border and the left border precisely, right? And then comes, as soon as this DNA is separated out, it cannot live on its own. It has to now require some assistance, right? So we have this Vir-D4 which attaches on one side. Now you see in the diagram there's the T strand. The T strand has this Vir-D2 which acts as a guide. Where is it taking to? It has to act as a guide to get into the plant cell. While this Vir-D2 is taking that small transfer DNA, there is a channel that is made between the Agrobacterium and the plant cell. Look at how beautiful these processes are. There is a channel. They build a nice tunnel so that the Vir-D takes the T strand, and suddenly come these Vir-E genes. What are they doing? Because once this strand enters the plant cell, it's a foreign DNA, right? It's a foreign DNA. The plant cell would like to chop it off. So these Vir-E genes, they coat the T strand. That coating protects the transfer DNA from being broken down by the plant cell. So see how beautifully those Vir apparatus has cut out a proper transfer DNA, and it is getting into the plant, making a channel, getting into the plant and protecting itself against the plant DNA, plant nucleases, sorry. And now it gets to the nucleus, it's making a journey. It's almost like an Argonaut, right? It's making a journey into the nucleus, and that nucleus gets through the nuclear membrane, gets into the nucleus, and it integrates into the plant. And you can see how each Vir element has helped, right from A to G to D to E, and then the cytoplasmic trafficking, and it becomes part of the plant. So this whole thing which I told you is the genetic engineering. And we haven't done anything here. Nature is doing it. So tomorrow when I hear a big crowd coming up to me with placards saying, genetic engineering, we want to ban genetic engineering. Sir, genetic engineering became part of it. A genetically modified organism is a task of nature. Whether you like it or not, it's been happening in nature. We just became part of it, right? So that's where I say that we need to kind of understand what's going on, right? Let's not down by the products of scientific research, but first let's understand what's going on in nature and look at how beautiful nature has been, or rather this natural genetic engineering in nature has set forth the fantastic mechanics of molecular elements, right? So yeah, I see that my voice is a bit intermittent. I hope it's okay with others, but let me know. But then let's get to what happened next. In 1983 itself, at Monsanto, we have these two fellow scientists, right? Robert Hirsch and Robert Fraley. In Monsanto, they were the first ones who standardized a beautiful process. It's called the leaf disc transformation. This is where any plant, now if somebody wants to become a plant genetic engineer, this is the process they do. They take a tobacco plant, tobacco is the model organism, and within 3 to 4 weeks, we cut out these discs from a tobacco leaf, right? And we literally put Agrobacterium on top of these leaves. So we take that petri dish and we took the Agrobacterium with whatever modifications we did. So what do we do now? In the tDNA, we remove the auxin and cytokinin and put a gene of interest. And we have an injured, when we cut out the tobacco leaf disc, the entire uniform injury around that will produce the phenolics, right? And the Agrobacterium gets it. Now, in the petri dish, along with the plasmid which we designed, we also keep an antibiotic resistance gene, which Robert Hirsch and Robert Fraley, with the leaf disc transformation, they used an antibiotic called kanamycin. So when you keep these leaf discs, we can get multiple shoots. So imagine from each leaf disc, in my personal experience, I can get up to 10 to 15 shoots, sometimes up to 25. So imagine from each leaf disc you have 25 plants, and if there are 10 leaf discs in a petri dish, you have 250 tobacco plants. That's the power of tissue culture. So these shoots which are coming out, if they are resistant to the kanamycin which I put in that plant medium, that means they have been transformed. And then I regenerate the plants, take them to the greenhouse. This is the first instance of a proper plant genetic engineering that was done in 1983 itself. So the applications were very fast. And that was at Monsanto. And in University of Wisconsin, Madison, we had John Kemp and Timothy Hall. One of my personal favorites because I worked with Timothy Hall at Texas A&M University. He was one of my mentors and I stayed in his lab for almost close to a decade in various capacities. My last postdoc was with Timothy Hall. He used to tell this story. What you see here, there is that what they did was Timothy Hall used to work with beans and he had this protein called phaseolin. And that phaseolin also had a gene switch along with it. So a lot of times when you do genetic engineering, there comes these objections. You are still using the viral elements, right, to drive the gene expression. So we had this promoter right in front of a gene of interest, which was called the cauliflower mosaic virus promoter. What if we remove that and put a plant-based gene switch, right? So this was the first time when they used a phaseolin bean protein with the phaseolin promoter, and they introduced that in sunflower. So here we have a plant protein from one plant being transferred into another plant. They call that resulting plant sunbean, right? So sunbean is a first genetically engineered plant where there was a horizontal gene transfer from one plant to the other. That was at University of Wisconsin-Madison. You see the title of the science paper that came. Phaseolin gene from bean is expressed after transfer to sunflower via the tumor-inducing plasmid vectors. And on the left-hand side, you see the New York Times actually putting out a small news item saying they put the bean protein into a sunflower. So these are the two path-breaking monumental works that went forth to harness. So where do I fit in? So what I've been doing all these years, right? I have been witness to the birth, the evolution, and later on almost 3 decades worth of work. And I had worked in, I started my life with peanuts. I used to have this big logos in my lab saying I work for peanuts. So they used to ask me, well, what are you hinting at? Is it the crop or your fellowship? But I couldn't complain. I started off as a junior research fellow with the Rockefeller Foundation, and then I became a fellow of Ciba-Geigy, right? So I started my life with peanuts. Then I came in 1997 as a Rockefeller fellow to Texas A&M University, started working with sorghum. Then I branched out to rice, cotton, wheat, tobacco, soybean, sugarcane, bamboo, and as a faculty member at Western Kentucky University, I worked with basil, right? Today I'm doing CRISPR gene editing. That's a whole new story. But within CRISPR gene editing also, our main tool in hand is Agrobacterium. So I'm editing out characters in hemp, cannabis, and peanut. I'm getting peanut back from where I started. Whole circle of life, right? So what did I do? The first thing I did was I took part in a project with cotton where I said my genetic engineering with cotton is environmental friendly. Environmental friendly as in there is a biocontrol agent called Trichoderma virens. What they do is they spray it on cotton plants. So in a cotton field you have organic farmers spray Trichoderma virens onto the plant because Trichoderma contains this enzyme called chitinase, and every fungus has an outer fungal wall called chitin. So they pretty much spray that so that chitinase cuts down chitin of the fungus and gets rid of the fungus. So what I said was, okay, let me talk more with the organisms. That's when we when our group found about Trichoderma virens. What we did was we isolated the endochitinase gene from Trichoderma virens, made it part of an Agrobacterium construct, and we engineered the cotton with that chitinase gene from Trichoderma virens. The promoters which drove that were in-house plant-based promoters. Of course, this publication still used 35S, but later on when we redesigned the vectors, we have a chitinase gene from a biocontrol agent now part of the plant of cotton. So that's why we call it the environmental friendly fungal resistant cotton. What did we do? With one gene that's integrated into the cotton, now we have resistance to two fungus. One was Rhizoctonia solani, which actually infects the cotton plant right when it's a seedling. Between the root and stem, it goes and causes what is called the lodging of the plant. So there you see. So it's like in the bottom of the slide, you see in a tray where the control cotton plants are all dead. We just placed it in a soil infested with the fungus, where the transgenic cotton is happily coming up in all its glory. Right. This is another fungus with one gene, a double whammy. We got rid of another fungus called leaf blight, Alternaria alternata. On the top row, you see all the cotton plants with the leaves where I put the agar plugs of the fungus. All the leaves are rotten, and you see the transgenic cotton leaves. Even after 2 weeks, the leaves are green as ever, right? Within the group, you also see one leaf rotting off. So this is another thing which we should be very worried about. I would be very worried if all the leaves are healthy. The gene should be Mendelian, 3:1, right? There should be still some plants which are susceptible, right? That shows that the gene is behaving the natural way, not like taken over the entire biological system. So that's another. We go deeper. Can we use gene switches which are so specific that they don't get expressed all over the plant, right? So we isolated some promoters or the gene switches from sugarcane, and we used them to transform rice. This is a very great work which went on at the Texas A&M AgriLife Research, a very amazing group. So we have people from this group who went on into the Corteva Agri-Sciences. Some of them are in Monsanto, some in Bayer. But they were all academicians at one point. But what did we do? We went so specific that rice has these sap-sucking insects. They go and suck the saps and stems. So we engineered these and specifically expressed at a place where the pest attacks, which is down to the sap in the phloem, protoxylem tissues. Right? So as you can see, when we transform the control with the reporter gene, nothing in the dirigent and osmotin, they are specifically being expressed in stem, not in the leaves and the roots. And within the stem, they just get expressed only in the protoxylem. So that's where the insects go and suck the fluids out of the rice plant. That's where, so we can be so specific. So it's not just genetic engineering, but what I would call a very tissue-specific genetic engineering. I'm also proud to have worked with Timothy Hall, who created that sunbeam, which I just showed you a little while back. We dug deep into that phaseolin. So this was where I did some bioinformatics work to decipher out more molecular elements. So that was one of my proudest moments, to be associated with one of the pioneer shifters in plant genetic engineering. Seen it all. So now today, I do work with CRISPR. CRISPR is very precise. It not only goes and inserts, but it inserts at a place where we want, and we can precisely edit out a gene. Again, I use Agrobacterium. On the left hand side, you see a hemp, which is all natural. On the right hand side, I precisely edited out a gene called phytoene desaturase, which shuts down the chlorophyll synthesis. And as you can see, that PDS edit for all these bleached out leaves. Right? So I continue to kind of be with the natural genetic engineering called Agrobacterium. Right? So we had a nature's genetic engineer hidden within plant tumors, and today I shared its biography with you. Right? So, and how precise this whole thing is, and how it brought about the thing called plant engineering. My basis for that was these two wonderful papers. One was called Agrobacterium Memoir. Right? That was what was written by Mary Del Chilton. And the other thing was called My Secret Life. Required reading. These are not technical papers. These are What excitement of science, it's an amazing read, these two. Someday I wish somebody chronicles entire biography with all the episodic history of all the scientists, right? So I leave you with the thought, it's not the fruits of scientific research that elevate a man and enrich his nature, right? But the urge to understand. If we understand life, that's where the greatest discoveries are all hiding, right, within us. And I would like to pay homage to my two great mentors. One is Professor G. Madhav Reddy, with whom I started my PhD. He was the one who took me to a plant molecular biology lab, right? And of course, Professor Timothy Hall, whom I met as a master's student in India, but I never dreamed that I would come and work in his lab, but I did. But these two people, these two great legends, are responsible for what I am today. And someday I hope to share their story also with you in a greater detail. But that's, we have to continue understanding life. That's where the greatest is all there. So I hope I conveyed a great story to you, and I hope you all get enthused to understand life in greater perspective. Yeah, and I invite any questions you might have to, further decipher this. Thank you. Thank you. Okay, the gene insertion by bacteria occurs in plants. Does it occur in animals too? Yes, it does. There are certain marine biologists who actually saw some proofs. I don't get the names of the organisms, but yes, they do. The natural genetic engineering occurs in animal systems as well. Yes, but that escapes my mind as to what specific examples I can give. Yeah. So have you heard of Dr. Suresh Patil, plant pathologist? Yes, Phaseolus vesicola. Yes, Phaseolin group is a fantastic group still. We are actually getting deeper into Phaseolin because Phaseolin as a protein, right, is very, very understudied. And I also have a pet project in bioinformatics where we look at the bioinformatics, the molecular evolution of Phaseolin. And we got into certain protein domains which actually we traced back to marine bacteria, right? So these are protein domains called cupins, and there are so many interesting biological functions, right from pathogen interactions to protein engineering and stuff like that. So what is coming in the near future? Great question. CRISPR has been a fantastic tool that is now being harnessed in plants. So now we can be so precise that within CRISPR there is an element called guide RNA. It not only takes you to doing something more precise. Now we don't even have to worry about introducing a gene from one plant to the other. What we do is we work with indigenous genes within the plant. Within the plant there are genes which are unwanted, and then there are genes which are not expressing to a level where they should be expressing. So this is what we now call as NGT, new genomic technology. It's not about genetically modifying a plant by bringing in a horizontal gene transfer, but we work with the plants, we work with the genes within the plants. That's where all the excitement is nowadays. And some of the good news is that even in Europe, where they were very averse to GMOs, now they are having a much more favorable reception to the CRISPR gene editing, right? But again, Agrobacterium is helping us to make those tools more and more precise. A day might come when we do what is called DNA-free editing, meaning we don't even use the molecular elements from outside, but we work with whatever is indigenous to the plants. So that's where that's where the future is. Can we just stay with the plants and modify them within their own genome too? And very precise. Yeah. Any other questions? And I hope this enthuses you enough to, you know, so you can modify the plant to make or less of something it not. Yes, that's exactly what we do with CRISPR. We get into the plant and see what's wanted more or what's wanted less, and we can edit that out. Yeah, precisely, and it's very precise because we now have this thing called guide RNA, which actually targets the genes which we want. Do you see the reluctance to using GMO retreating into industry? Yes. The way that thing will retreat into history is when more and more of scientists get out there and educate the public. Citizen science is what is really the need of the hour, because a lot of it is misinformation, right? So if we go and tell these science stories, right from kids at the kindergarten level, to make them excited about the technology and give them the wherewithal to see that how beautiful the nature is. If you make the beauty come out, that's when they sit and listen to a favorable mind, because we are spoiled by all the Frankenstein stories, right? But what about the wonderful beauty in nature, right? That is, I think, what is required. Fantastic. I'll be glad to share as many stories as possible. I have a whole stack of stories, many, many exciting as this. So, yeah. Yeah, that's another thing that's going on in the political turmoil, getting away from WHO and getting into NIH. But I hope that saner elements come into the picture and support understanding. Yeah. Yes, food crisis can be solved with this technology. That's another great area. The arable land is going down. And if you really look at it, the next 50 years, the next 50 to 100 years, we have to produce as much food as we have for the past 10,000 years. 10,000 years ago was when agriculture started as a practice. But the amount of food that's being produced, the same amount of food needs to be produced in the next 100 years, and we don't have any cultivable land to get that. So what we are looking at is high yielding varieties which can be grown right in your own backyard, right? Tissue culture and uh, natural genetic engineering is what can take up the challenge and integrate into our lives. Yes. So more food for us. Less food for wildlife. I won't particularly agree with that, because we can even ensure that the wildlife is also properly fed. Right. So it's we can help each other. Just like Agrobacterium went into the plant and made its own octopines, and it is helping harness. It's an endosymbiont, right? Otherwise, it would have considered itself as a pathogen, and we might be busily trying to get rid of it. But no, we harnessed. We made that an endosymbiotic relationship. Otherwise, what the plant was getting was just crown gals. Now we have the Agrobacterium happy, and it is making our food for us. So the more vulnerable we may be with less and less diverse crops. Yes, that's true. Because now, since we are indigenously working with each species, we need not worry about a gene of one plant jumping into the other, right? So the precision is what is, yeah. Oh yeah, wonderful. Slimy but satisfying. Yeah. So the future of genetic engineering, and Tony Stark is asking me that question. The future of plant genetic engineering is make more nutritious food which can protect itself from nature. And thank you so much for your kind best wishes. We need that, right? So yeah, so I can't emphasize enough. It's not the fruits of scientific research that elevate a man and enriches nature, but the urge to understand. So yeah, the sky is the limit. Chocolate and coffee from petrochemicals. Why not? It's harnessing nature for what we need. Yeah. Okay, if there are no more questions, then thank you again. It's been a wonderful. Uh, thank you so much. And I wish I would come back. Yeah, I'll be back with another talk, I guess. So we'll meet now. It's and I really enjoy my time here. So thank you, Stephen, for giving this great opportunity.